The enzyme protection assay enabled to study the extent of Staph aureus internalization and its ability to survive inside adherent cells, as well as the efficacy of antimicrobial compound. The improved enzyme protection assay greatly simplifies technical handling and enables to recover internalized bacteria from weakly adherent cells and cells in suspension. Demonstrating the procedure will be Josselin Rigaill, a medical doctor and PhD student from my laboratory.
Two days before the infection assay, seed A549 epithelial cells at a density of 2.0 times 10 to the 5th cells per milliliter per well of a 24 well plate and incubate the cells for 48 hours at 36 degrees Celsius and 5% carbon dioxide until they reach 100% confluence. On the day of the assay, remove and discard the spent culture medium from the 3 wells dedicated for counting the A549 cells and add 1 milliliter of complete infection medium containing 5 micrograms per milliliter of Hoechst 33342 and 1 microgram per milliliter of propidium iodide. Incubate the cells for 30 minutes, then using a wide-field fluorescence microscope, count the number of cells and calculate the cell viability. To prepare the bacterial suspension, dispense 25 milliliters of complete infection medium in a tube and pre-warm it at 36 degrees Celsius. Next, adjust the Staphylococcus aureus suspension to an OD of 0.5 in a complete infection medium using a cell density meter. Then, prepare 20 milliliters of bacterial suspension for cell inoculation.